So hello students, how are you all? So hope you all are doing great. So, so all those students who are appearing for this year NEET, of course you all have to have some time to appear for NEET, but this is the last week. So just keep on tracking if you have your elder brother, sister. So just those who's any of brother, sister or in the family, anybody appearing, just give me a thumbs up so that I can understand. So just you can just observe what are the feeling when NEET is very close to each for the student, right? And you also have to manage those feelings. So, but anyways, during the entire 2 year, you will understand how to manage all those feelings. So till then, you have to keep on continuously working hard, make your subject strong so that in the end you don't have to worry about exam at all. So let's start with our today's topic. So we have already talked about, okay, so I can see many of the students, some of their brother or sister is appearing for NEET. Great. So my voice is clear, then just give me a thumbs up and when we will start. So here in last class, as we have seen that, in last class we have already talked about many eukaryotic cell organelle, and that included, those cell organelle included till mitochondria, I think we have completed. Now today we will start with the chloroplast. So here it is. So we will today start with the chloroplast. Now chloroplast and mitochondria, they are kind of of course they are functionally different, structurally also there are differences, but they have some basic commonality, right? We have already seen that mitochondria is two membrane structure, means there is an outer membrane, there is inner membrane. If you see here chloroplast, now this chloroplast is also having a outer membrane and it is also having an inner membrane. Outer and inner membrane is present here, and in between there is intermembrane space. Same thing was present in mitochondria also. If you see outer membrane, inner membrane, intermembrane space. Now if you see inside the mitochondria, the semi-fluid substance was called matrix. Here that semi-fluid substance is called stroma. In mitochondria, we have seen that the inner membrane was folded, forming finger-like projection called cristae. Now here they have a separate membranous structure called thylakoid, right? Several thylakoid will come together to form granum. So let's first understand the arrangement of granum and thylakoid. So inside is the stroma. Now imagine a 5 rupee coin, right? 5 rupee coin, if I draw that 5 rupee coin, let's consider this is a 5 rupee coin. Now if you place several such 5 rupee coin one over each other, let's say you have 5 rupee coin. So if you keep all of them one over each other, right, on the top of each other, will it form a stack, right? So see here, the stack is called grana. The stack is called grana and the single one, the single unit is called thylakoid. Understood? So here again you can see these are individual stacks. These are individual thylakoid which together one over each other is forming a stack called granum. So granum is the stack, individual is called the thylakoid. Now thylakoid is not exactly like a 5 rupee coin. Actually thylakoid is hollow inside, right? Means if I draw the structure of individual thylakoid, so thylakoid has a outer membrane and inner lumen. If individual thylakoid I draw, so thylakoid has a outer membrane and this membrane is called thylakoid membrane. And inside is the thylakoid membrane and inside is the lumen called thylakoid lumen. Is it clear? It is just like it is surrounded by a membrane and inside is the space. The space is called lumen. So this point is very much clear that thylakoid has a membrane and thylakoid has a lumen. Several such thylakoid one over each other will form grana. Now inside the stroma there can be multiple grana. For example, let's say this is one grana, this is another grana, this is another grana. So there can be multiple grana and they are connected to each other. Let's say this one is connected, this one is also connected. So these grana are connected to each other and that connection is called stroma lamellae. That connection is called stroma lamellae. As you can see here, this is the stroma lamellae. So the connection between the grana is called stroma lamellae. So 2-3 key important points are there. The first one, they have outer membrane, they have inner membrane. They have a intermembrane space. Inside semi-fluid matrix is called stroma. Inside the stroma they have membranous structure called thylakoid. Individual thylakoid has an outer membrane called thylakoid membrane and inner lumen is present. Several such thylakoid stack together to form grana. Several such grana will get connected to each other with the help of stroma lamellae. Till here everything is clear, right? Now, in addition to these, inside the stroma, not shown here, but they can also have ribosome. In mitochondria also have we seen ribosome? Yes. So there will be ribosome. Then there will also be a circular DNA called plasmid. Circular DNA called plasmid. Is it clear? See, in cell, two things you have to understand which is very important. One, the structure of cell organelle, and second is the function. Now function is easy to remember. Main point is the structure. So if you remember the structure, then everything is easy here. So this is all about the structure of chloroplast. So just see all the part. Now I am again going to draw the structure with your help, right? So let's draw the structure. In this way we can memorize it also. So let's see. And I will draw and you have to label it, right? You have to label. I will write A, B, C, D and you will label it. So now let's start with this labeling over here. So first I am drawing a chloroplast. So chloroplast will be a double membranous structure, right? Now, so here as I said, you have to label it. So I am putting A and B and C and you have to tell me what is A. I am telling what is B. Then the third, what is C. All of you start writing. You have to write like this. A is this, B is this, mention A dash, whatever it is. We have just seen. If you answer it, I will understand. Yes, correct Khalil, in this way. Ram Babu, fine. Yes, in this way you have to write. Shanmukh, very good. So continue writing and I will be drawing inside, right? Then apart from C, there is another D. Tell me what is D. Then We will further draw. Write for A, B, C, D, and then you can continue with other labelings. So let me see how many of you do correct. So A, B, C, D, then E, label E, then label F, then label G, right? Label G, then label tell me what is H, then inside and this one is I. So this is all you have to label. Now as I can see, many of you are answering. Okay, so Janu is telling B is the inner membrane. Okay, then Vijay is telling E is the thylakoid. E is the thylakoid. Ritika is telling E is grana. See, mind it, E is grana or E is thylakoid? E is grana or E is thylakoid? What is F and what is E? Right, Sandhya is telling A is outer membrane, Reddy is telling B is inner membrane. Okay, Tanuj, A is inner membrane. A is inner membrane. E is grana. Okay. E is grana or E is thylakoid? F is grana. Okay, B is very good. So many of you, Ramachandran, very good. Divya, Bhushan, Harsh, Akhil. Yeah, Surya, answer. I can see you. Reddy is telling I is plasmid. Okay, H is what? H, G is stroma, F, very good. So H is ribosome. Sheikh is telling H is ribosome. F is grana. Very good. So now once I will just quickly tell you again. See why I am giving you in this way, because this is exact how you will be asked question related to NEET. Labeling of diagram is asked in this way in NEET, right? So all of you, if you want, you can have a screenshot and then you can practice writing all these labelings, right? So now if I go with the proper labeling, so here A is the outer membrane, which all of you have said correct. A is the outer membrane, B is the inner membrane. So there is no confusion between outer and inner membrane. Now in between outer and inner C is the intermembrane space. Intermembrane space. Then inside. So outer, inner, inter. Now let's get inside. So inside is D. Now D don't confuse with matrix. D is what? D is the stroma. Few of you have written it as matrix. D is stroma. Matrix is present in mitochondria. So it is a stroma. Let's get to E. E is what? E is thylakoid. E is thylakoid. The individual stack is called, this individual stack is the thylakoid. This individual is called thylakoid and this entire stack together is called grana. It is called grana. Now connecting to grana is the stroma lamellae. And finally, you have this ribosome. Can anybody tell me what kind of ribosome it will be? 70S or 80S? Write in the chat box quickly. What kind of ribosome will be here in the chloroplast? Will it be 70S or 80S? And finally, I is the plasmid. Plasmid is the circular DNA which is also present in bacteria. So just like mitochondria, it also has its own ribosome and it also has its own plasmid. Yes, waiting. Okay, Tanish has 770S, Neeraj 70S, then okay, few of you are writing 80S also, but maximum are writing Reddy 70S, okay, Shri 70S, 70S, fine, right. So majority of you are writing 70S, some of you are writing 80S. See, remember, ribosome present inside mitochondria or chloroplast are always 70S. They are always 70S. Inside mitochondria or inside the chloroplast, they are always 70S. See, so every time, see, this is a very simple trick to memorize the diagram. First, you read the diagram and then try to draw it by yourself and label every component which is given in the diagram. And in this way, you can easily recall and remember everything. Is it clear? Okay, so now let's move ahead. So if we move ahead, so here, few questions about the chloroplast, but before that, let's see the function. We haven't talked about the function. Chloroplast is present in plant, so it has the major role in, chloroplast has the major role, that is if we talk about the function, so it has a very important role to play in photosynthesis. Why it has an important role to play in photosynthesis? Because chloroplast has pigment. What kind of pigment? So they have photosynthetic pigment. Now if I ask you, where is this photosynthetic pigment present? Where is this photosynthetic pigment present? So they see this thylakoid membrane. Thylakoid membrane. On the membrane of thylakoid, photosynthetic pigment is present. Question is asked in previous year in NEET that where is pigment present in chloroplast? So answer will be membrane of thylakoid. If you get option lumen of thylakoid, no, only membrane of thylakoid. So very good. So understood. So the main function of chloroplast is photosynthesis. How? Because they have pigment for photosynthesis. Where is the pigment present? Membrane of thylakoid. Clear? So now function is we have seen. Now we can see some of the questions. So question time, all of you get ready. Now you have to answer. So first question says that the primary function of chloroplast is to. Just I have told the function. Just I have told the function. Only, all of you, only you have to write the answer. No other message in the chat box. Only the function. Only the answer you have to write. And here in this question, what is the primary function of chloroplast? We have just seen. Yes, Tanish, Yashwant, Sheikh, Lasya, Ganesh. Very good. Very good. Brilliant. So all of you are telling correct answer. None of you have given any wrong answer. So very good. Means the primary function of chloroplast is to carry out photosynthesis. Very good. Then next question. Chloroplast contain which pigment that is essential for photosynthesis? Now this is a very common sense question. Common sense question. You all must have studied in your previous class also. Yes, again Arjun B, Rao B, Meena B, Pinky B. Very good. Very good, you are answering B. Pranav, just write the answer. Just write the answer. You have to write only answer A, B, C, D. Okay, so chloroplast contain which pigment? So which pigment Green pigment, what is the green pigment? So all of you must have studied chlorophyll. Now, till now you must have studied only about chlorophyll, but now chlorophyll will not be alone. Now we will see in photosynthesis, when we will study, we will realize that chlorophyll can be of several types. It can be chlorophyll A, it can be chlorophyll B, it can be chlorophyll C, it can be chlorophyll D, right? So chlorophyll is of several types. So if I ask you in plant what is present? In plant what is present? So in plant present is chlorophyll A and B. Okay, so now let's move ahead. Another question. So this already we have, this is already done, chlorophyll. Now next question. Chloroplasts are found in which type of cell? In which type of cell chloroplast is found? So chloroplast is found in a cell which is photosynthetic. Now which cell is photosynthetic? Very simple. Very simple here. What is the answer? Yes, Khalil is answering 3. Sanvi 3. Jyoti C. Yadav C. Very good. Very good. So it is the plant cell which perform photosynthesis. So chloroplast will be present in plant cell. Will chloroplast be present in bacteria? Answer is no. Chloroplast not present in bacteria. Will chloroplast present in animal cell? No. Fungal? No. Right? Fungus? No chloroplast. Chloroplast is present in plant cell. Next. So now once we have completed the chloroplast, now we will move on to the next cell organelle, and that is cytoskeleton. Now no need to use the chat, just stay away from the chat and let's understand this. So cytoskeleton, see, it is made up of 2 words. Cytoskeleton means cyto and skeleton. So cyto means cell and skeleton has a role in support. So structure which provides support to the cell is called cytoskeleton. Just like bone is the skeleton in our body, and bone is responsible for providing shape to the body, help in movement, right? So in the same way here also the cell get a particular shape because of presence of cytoskeleton, and it also helps in performing certain function of the cell. So first, cytoskeleton inside the cell is of 3 types. Now as I can see, if it is not very clear, so is this diagram clear or should I redraw here? Just quickly tell me. Quickly tell me, should I redraw the diagram or is it not clear to you? Okay, so I will redraw it, right? It may not be very clear to you because of the white background, so I will draw it for you, right? Okay, yes Khalil, I am redrawing it, fine. So here see, this is the cell which is drawn here. Now inside the cell there will be a nucleus. Now apart from that, cell will have, see, some thick network like this. So these are one of the network. Then further they can also have some small network like this, comparatively thinner. Like this, and finally the outermost is like this. Now see, so basic, if you see here, you will see that there are 3 types of cytoskeleton present in the cell. You can see one is the thick one and surrounding. In the red color, what you observe? Surrounding the nucleus, and which one is appearing to be the thickest? So you will say, sir, the red thread is looking thickest, right? And then there is surrounding it, and finally the outermost. So the red one which is present near it is called as the microtubule. This one will be called as the microtubule. So one of the cytoskeleton is microtubule. First one is microtubule. Then comes the second one. Now second one, you can see here, this is the thinnest one, and this will be called as microfilament. And this outermost, this one is called intermediate filament. So first, second and third. Now here, see, you have to simply remember that there are three types of cytoskeleton. One is called microtubule, another is called microfilament, and another is called intermediate filament. Now, thickness wise, if I write, so this you can note it down. Microtubule is the thickest one. Then after that comes the intermediate filament. And which one is the thinnest? The thinnest one is the microfilament. Remember, thinnest one is the microfilament. These three, according to their size, according to their diameter, this is the order. So just you have to remember 2 points. Question is asked. This is a very small topic. Only 2-3 types of question is asked. First is, what are the 3 types of cytoskeleton? So your answer will be, it is microtubule, intermediate filament, and microfilament. Then question, if I ask, which one is the thickest? So the thickest is the microtubule. And which one is the thinnest? The microfilament. And remember, intermediate filament, name itself is intermediate, means it has an intermediate thickness. What will be its function? So its function is that it provide shape to the cell. And second important function, it helps in locomotion. Third important function, it provide support to the cell, right? It provides support. So function is 3 function. Remember, these 3 are the function. So just the type of cytoskeleton and function of cytoskeleton, nothing else from here, right? If you just remember this much, all question in NEET you can easily answer, no problem, right? So just keep it in mind. All of you understood it? So now let's move ahead. So this cytoskeleton is one type. Now if we see the cell has outgrowth. Now if you can remember prokaryotic cell have cilia and flagella. In the same way, here you can see prokaryotic cell do not have cilia, it has pili and flagella. Similarly, here is cilia and here is flagella. Now if you see here the diagram, this is the cell, and from the cell is coming out the flagella, right? Apart from that, this is another cell, and then you can see small thread like pili is present here, small thread like. Now if I ask you which one is longer, just answer in the chat box. Which one is longer, Flagella is longer or cilia is longer? So, very easy to answer. Flagella is longer and cilia is smaller. Smaller. Very simple. So, flagella is longer and cilia is smaller. Then, here, if I ask you which is less in number and which is more in number, if I ask less in number, so you will say flagella. So, flagella is less in number and cilia will be more in number. More in number. Now, third important point, flagella helps in locomotion. Flagella helps in locomotion. Cilia helps in locomotion as well as attachment, and in some case it also helps in movement. So you understand the difference. Flagella and cilia, one is longer, another is shorter. One is less in number, another is more in number, right? Another very important point, flagella is less and the movement of flagella is independent, while cilia all move together, right? Cilia all move together. So here, let me draw this one diagram. See, in the same cell, if I draw, so let's say this is the cell. So if I draw cilia, several small structure is present here. Whereas if I draw flagella, so flagella is a single long structure. So very clearly you can see the structure. One is longer, another is smaller. One is numerous, another is very few. Clear? So flagella and cilia. So externally they are different in length, but internally they are exactly the same. Internally they are exactly the same. Now one more point here, and in NEET several time question is asked. The first question, you have to tell true or false statement. First statement says that flagella is present in both prokaryote and eukaryote. Both prokaryotes and eukaryotes. Now tell me whether it is true or false. Whether it is true or false. The statement is flagella is present in both prokaryote and eukaryote. True or false? True or false? Khalil is telling true. Rani, Harsh is telling true. Okay, so some of you are also writing false. So have we studied about flagella in prokaryote or not? We have studied about flagella in prokaryote also, and we are studying about flagella in eukaryote also. So what we will say, flagella is present in both prokaryote and eukaryote. It is true statement. It is true statement. Simple. Flagella is present in both prokaryote and eukaryote. Here another statement, if I write cilia present, cilia is present in eukaryote, flagella in pili in, not flagella, cilia is present in eukaryote and pili is present in prokaryote. True or false? Tell me. True or false? Waiting. Cilia is present in eukaryote, but pili is present in prokaryote. So, answer is Okay, I got 2 false answer. Okay, Dandelion is telling true. Ritika true. Lot of false. Arjun true. Many of you are writing false, true, false. See, again recall what we have studied in bacteria. Bacteria was having outgrowth, 3 outgrowth bacteria was having. One was flagella, another was pili, and third was fimbriae. So bacteria or prokaryote have flagella, pili and fimbriae. Was there cilia present in prokaryote? No. Cilia is present in eukaryote. Cilia is present in eukaryote. Pili is present in prokaryote. Right? P for pili, P for prokaryote. If you get confused, remember this. Pili is present in prokaryotes. Cilia is present in eukaryote. So this statement is also true. Understood this? Now, further if we see, so these 2 statements are true. Now if I say further, then the membrane extends itself and it forms cilia or flagella. Remember, it is something like this. See, if I am drawing the structure of cilia and flagella is exactly the same. It is exactly the same. Structurally, they have no difference. Function is different, but the structure is the same. They are surrounded by a membrane. And basically, this membrane is nothing but the cell membrane, right? The outer exposed part is called the axoneme. And this part is called the basal body. So, flagella, see, if I simply write down that flagella in eukaryote or cilia in eukaryote consists of 2 parts, axoneme and basal body. Is it right? If I say axoneme is surrounded by cell membrane, I can also write this one. Axoneme surrounded by cell membrane. Surrounded by cell membrane. Whereas no membrane is, if you see basal body, no membrane in basal body. No membrane in basal body. So overall, can we write like this? If we write in the form of equation, if we have to write then equation on this concept, so can we say axoneme plus basal body forms flagella or cilia? Is it right or not? You see, this is the membrane extends itself, and this is the basal body, and this is the axoneme. So axoneme plus basal body, they together form the cilia or flagella. So this you remember, because this concept will help you to understand, because now onward whenever I will say axoneme, so you should understand that I am talking about the exposed part of flagella. Whenever I will tell basal body, so you will understand that it is present inside the membrane. So axoneme will be the exposed part, and the basal body is in membrane. Now, is this membrane bound? So no, it is not membrane bound. And here it is surrounded by membrane. Surrounded by membrane. So till now everything is clear. If I say flagella has 2 parts, so now we will not stop here. We will see axoneme, structure of axoneme and structure of basal body. So if I say structure of axoneme, you will say that, sir, you are talking about the exposed part of the flagella. When I say basal body, you will say, sir, you are talking about the part which is present inside the membrane. So let's see the structure one by one. So here, first of all, this part is called centrosome or centriole. So here, if I say these are the two different structures. So if I say here this part, so this part represent the axoneme structure. Now, as I said, I will say axoneme of flagella. What do you mean by this is the exposed part? Now let's imagine that this is the flagella. Let's consider this is the flagella. Now if we cut this flagella and if you observe it in this way, this side if you see, then this will be the arrangement which is shown here, right? This is shown here, axoneme. If this is the flagella and if you cut from here and you see it in this way, if you see it in the diameter, then you will get this arrangement. Now, so this appear to be a very complex diagram. So let's break it one by one. This diagram is given in your NCERT. The diagram is given in your NCERT. So here, if we see, this is the axoneme, structure of axoneme. Now, if you see here, what are these circles present? You can see there are several circles. In some case, they are present in pair, as you can see here, and somewhere it is present not in pair, but they are single, right? So these circles are called, these are basically microtubules. Now you will say, sir, microtubules we have seen in the cytoskeleton. Yes, it is the same microtubule, right? So you will say that microtubule help in formation of axoneme. We will also see it is also present in basal body. So microtubule help in formation of axoneme and basal body. So here, now if I say how these microtubules are present in pair, can you say that? It is very much clear that here they are present in pair. So when they are present in pair, can we call them doublets? Can we call them doublets? So answer is yes. So these are doublets. Now my question, how many doublets are there? So if you count starting from here, 1, 2, 3, 4, 5, 6, 7, 8, 9. So how many doublets are there? 9 doublets. 9 doublets, right? Each doublet is having how many microtubule? Each doublet is having how many microtubule? 2. 2. So what we will say? These 2 into 9. Can we say peripheral microtubule? Peripheral microtubule is 9 into 2, which is 18. Can we say that? Understand this. This is peripheral, no? Surrounding is peripheral and this is the central part. So now if I say peripheral microtubule, so you will say peripheral microtubule are 9 doublet. 9 doublet means 9 into 2, that is 18. In the center if you say, see in the center it is not doublet because they both are not attached, they both are separate. See, they are separated with help of a small linker, isn't it? So center is not doublet, center is singlet, right? So we will call it here central singlet. Central singlet is how much? 2 central singlet. So 2 into 1 is equals to 2. So here what we can say? We can say here that the peripheral microtubule is 18. So can we say here 9 plus 2, right? This arrangement is called 9 plus 2, where 9 is the peripheral. Peripheral means which is surrounding. Peripheral doublets. How many peripheral doublets are there? 9. And each doublet have 2. So peripheral doublets have how many microtubule? 18 microtubule. Whereas there is 2 central singlet. 2 central singlet means what? 2 and singlet means 1. So total how many microtubule? 2 microtubule. So total how many microtubule? So 2 plus 18, 20 microtubule. So now here question will be asked. Very simple question. Which of the following is the microtubule arrangement in axoneme? What you will say? Is it 9 plus 2, 9 plus 0, 9 plus 3? So answer will be 9 plus 2. Now question is asked, 9 plus 2 is the arrangement of what? So you will say arrangement of microtubule. Now 9 stand for what? So you will say 9 stand for the peripheral doublet. So now if I ask you peripheral doublet has how many microtubules? So you will say 9 into 2, 18 microtubule. Then I will say what is this 2 representing? So you will say 2 represent the central singlet. So how many microtubule in central singlet? So you will say 2 into 1, that is 2 microtubule. So what is the total microtubule present in the axoneme? So you will say 18 plus 2. So that is 20 microtubule. Is it clear? So now can we say that the axoneme has in total 20 microtubule? Out of that 18 is peripheral and 2 is central. The peripheral microtubule is present in 9 doublet and central is present in 2 singlet. So this is the arrangement of axoneme, which is called 9 plus 2 arrangement. 9 plus 2 arrangement is the arrangement of microtubule in the axoneme. Now this I haven't talked about because here no 9 plus 2 arrangement. Here we will see 9 plus 0 arrangement. Right? So 9 plus 0 arrangement is for centrosome or centriole and 9 plus 2 arrangement is for axoneme. Right? For axoneme. So this in detail we will see tomorrow. Right? So just I will leave it here today by writing this one important point. The arrangement of axoneme. Axoneme arrangement is 9 plus 2. And if I say arrangement of centriole, so it will be 9 plus 0. So we have today discussed in detail about 9 plus 2 arrangement. This we haven't talked about. So in the next class we will see what is 9 plus 0 arrangement and is this arrangement how it is related to basal body. This we will do in the next class. So you need to go back, you need to read NCERT, revise NCERT, and most importantly, today you go back and see all the diagram given in NCERT of all the cell organelle. Because if you master diagram in this chapter, then most of the question you can easily answer. So that is it for today. So all of you have a very good day ahead. Enjoy. Thank you all.